Welcome to Decoding Healthcare Research. Join us as we delve into the behind-the-scenes world of groundbreaking research and the dynamic healthcare industry, interviewing top paper authors, engaging experts on industry-related topics, and exploring medical subjects that affect our daily lives. And now, your host, Dr. E.F. Rain.
Welcome to Decoding Healthcare Research. I'm your host, Efrain Riveros, Dr. E.F. Rain. I'm an associate professor of anesthesiology at the Medical College of Georgia. And today we are going to talk about aging. But this is not the regular topic that we normally touch on when we talk about longevity. This time we are going to focus on the application of data analysis and very sophisticated tools to start talking about biological age and also chronological age. And we have an expert and someone who is doing research in this field. And I am particularly impressed with the research products that his group is producing. So we have here Dr. Adib Johnson. He is the Director of Research and Innovation at Tally Health in New York in the USA. So first of all, welcome, Dr. Johnson, and thank you for sharing this time with us.
Thank you so much. It's a pleasure to be here.
Thank you. So when we talk about aging, this is a concern for everybody. We tend to be more concerned usually after the fifth decade of life, which is my case. But the process of aging goes hand in hand with a series of physiological changes. So the first question I want to ask Dr. Johnson is, for how long have you been doing research related to aging?
Yeah, that's a really good question. I actually, from a very young age, have been obsessed with aging, and I really don't know why, but I remember in high school knowing that I wanted to go into the biomedical research space to learn more about aging. And as an undergraduate, I actually worked in a mosquito laboratory in the entomology department under a wonderful professor, Dr. Michael Reilly. And I did aging research in mosquitoes. In particular, we were the first group to show that if you calorically restrict mosquitoes, they live longer, just like many other animals. And we published that. And we also tried giving a variety of molecules to mosquitoes to see if it would impact their longevity. And that was my first foray into that space. And I want to say I started that research around 2008. So it's been a handful of years since then.
I see. And we all know that as not everybody ages at the same speed. And you can see someone in his 50s who looks way younger than someone the same age. So that means that the chronological age probably doesn't tell the full story. So in your paper, you start mentioning the concept of biological age. What do you mean by that?
That is one of my favorite questions. So when I think of biological age, I immediately think of this abstract, intuitive concept that we all understand, where just as you described, if you see two people that are chronologically age matched, let's say you have two people that are 75, one person can climb up Mount Everest or run a marathon, and another person might be struggling with everyday tasks like cooking or going to the grocery store. Most people understand that these two individuals have aged differently. One seems much more youthful for the chronological age than the other. And this really gets at the heart of biological age, which is how we're aging, not just externally, but internally and functionally as well. Whereas chronological age is just how many times we've been around the sun or how many candles you put on your birthday cake. And I would actually say that, because we'll probably get into this some more, biological age at this time is not something that's clearly measurable in a very obvious way. It's more of an abstract idea. But what we have instead are indicators of biological age. So metrics that provide insights or unique windows into how we or an organism are aging on a biological level. So I don't know if that distinction makes sense about biological age as a more philosophical concept, and an indicator of biological age is something that's very concretely measurable.
Yeah, that's true. And actually, is sometimes it's difficult to to know where to start. What metabolic markers we can take, what genetic markers we can use. And I guess one good starting point are the 9 hallmarks of aging, published in 2013 by Lopez Otin, and two of them caught my attention: the epigenetic changes and genomic instability.
Yeah.
And those two in particular open a new window to understand aging. And I think you put it very nicely in your paper. And I would like you to explain our audience, for people who are not experts like me, what is epigenomics, and how can you incorporate that into the concept of aging?
Yeah, so as you described, there are known hallmarks of aging. They actually, very recently, the same authors came out with new work, and they've expanded it to 12 hallmarks. So now they've added chronic inflammation, dysbiosis, and another to make the list a total of 12 versus 9. And epigenetic alterations is a fundamental one. It's a primary hallmark of aging. And when I say primary hallmark, I mean it's thought to underlie other types of dysfunction. So something like chronic inflammation is a result of more fundamental changes that happen. It just doesn't just happen spontaneously. Many other things have to occur to get to a point, such as the accumulation of senescent cells that are secreting inflammatory factors to start to see inflammation in tissue. But what is epigenetics? Epigenetics basically describes mechanisms that affect gene expression without altering the underlying sequence of DNA. And a really common example of this is DNA methylation, where you add or remove a chemical methyl group to a cytosine base preceding a guanine, a CpG site, in the genome. And there are about 30 million sites that are eligible for DNA methylation. And typically speaking, when you add a chemical methyl group to one of these sites, you block gene expression. So let's say you hypermethylate a promoter of a gene. A typical effect would be that expression of that gene is suppressed. Now, that's not always the case because DNA is complex and there are many different regulatory elements in our genome. But that's just one type of modification to DNA that can impact gene expression without affecting the underlying sequence. Other examples include histone methylation, histone acetylation, and others. But these are all, we're all talking about modifiable chemicals that can be attached to something or removed from something.
So we could say that the problem, if you will, starts at the epigenetic activation as a result of abnormal patterns of methylation and demethylation, and then the expression is biochemical and probably inflammation and everything that we know under the concept of inflammation, right?
Yeah, I would say because aging is so complex, we still don't fully understand it and know the sequence of events. But we do know that some changes have to be more fundamental than others. So for example, if there's a mutation that occurs, let's say there's UV damage and a mutation occurs in a cell where a cytosine base ends up getting swapped out with another base. Well, that could in turn impact methylation of that site, which could in turn impact gene expression and protein expression. And then if that protein happens to be involved in an important process like managing the immune system or fighting pathogens, that could be affected. So that's how you kind of scale from more fundamental damage type changes to these more cellular and physiological manifestations of aging that we see.
Excellent, excellent. So now, if we understand that the epigenome is important, probably upstream. The ability that we could have to measure these methylation patterns can, in one way or another, be used as a tool to kind of anticipate and predict the biological age. Is that a good way to describe it?
Yeah, I would say that we know the methylome gets dysregulated with age. I mean, unfortunately, almost everything gets dysregulated with age, whether we're talking about the transcriptome, the proteome, the metabolome, or the methylome. But we do see, when we look at a cohort over time, whether it's cross-sectional or longitudinal, it's very clear that certain sites of the genome are very likely to become hypermethylated with age, so increase in methylation, while others become hypomethylated, which means they lose methylation. And obviously there's individual variability, but these changes are so predictable on a population level that you can actually quantify the degree of methylation and do a really good job predicting how old somebody is. And that's where you get these epigenetic aging clocks that are able to predict age with very high precision. But we don't want clocks that are perfect, because those would just be forensics tools. And there's actually room for that. You can imagine a variety of scenarios where you might want to analyze someone's epigenetic age for forensics. But at telehealth and in the aging community, we're interested in these tools as aging biomarkers. And so higher quality clocks, what we call next generation clocks, there's an age prediction that's going to deviate from chronological age. And that difference between chronological age and epigenetic age is called delta age. So if somebody has a delta age of plus 5 years, that means their epigenetic age is 5 years higher than their chronological age. Someone has a delta age of minus 10 years, their epigenetic age is 10 years younger than their chronological age. So high quality clocks, that difference, that delta, significantly associates with things like mortality risk over time, things that are known to be longevity promoting, like increased physical activity, better diet quality, having a more manageable life in terms of stress and social engagement and fulfillment. The first generation clocks were really just trained to estimate chronological age and don't associate as well with these longevity factors that we care about. But these next generation clocks do a much better job, and I would argue do provide insights into aging on a biological level through the lens of DNA methylation.
That's excellent. So we can understand these aging clocks are as models, right? And these models have been perfected over time as they incorporate more variables, I assume. And there is a component of prediction, as you mentioned, but is there also a component of inference so that you can understand better the process of aging based on the model itself?
Yeah, so we actually just published it just came out a couple days ago, a brand new paper in the journal Geroscience. It's open access if anyone wants to read it. And to your point, exactly what you're asking, we not only created a new clock and showed the methodology for that clock, basically a way to maximize signal and minimize noise, but we also looked at the methylation sites, how they changed with age, and what the associated processes were to derive biological insights into human aging. And we saw really, really fascinating trends in terms of how methylation increases or decreases over time, the types of pathways that are linked to these methylation sites that significantly change. We saw immune pathways, for example, which is not a surprise. And we also saw that there are clusters that gradually change with age. There are others which only shift very rapidly one other we found that shifts very rapidly at the end of life. And so you can absolutely use these tools to learn more about the biology of aging and what's going on a molecular level. And because it's so complicated, when we're dealing with massive amounts of data, we have to use data science tools like machine learning, regression models, and other approaches to gain these insights.
Yeah, and that's at the very center of the paper. Do you mind explaining in simple terms what is a machine learning algorithm applied to these clocks?
Yeah, so usually you can make an aging clock out of anything, by the way. There are aging clocks for proteins and genes and metabolites and other types of molecular data. But when we say epigenetic aging clock, we're almost always talking about a specific machine learning model, which is a regression type model, either elastic net or lasso. Those are the most common approaches. Now, there are people that use more deep learning type approaches, but I would argue that those are not superior in terms of age prediction, in the context of you have one type of data and you're trying to optimize this prediction. But a human couldn't go and look at, let's say, 8,000 people, and you have methylation epoch arrays for all of them, which measure almost 1 million CpG sites, and then go, okay, let me see which of these change with age. And then I wonder if I could combine these in a way mathematically that I would be able to predict someone's age. And then also the difference between my predicted age and their actual age would correlate with their health and lifestyle. That would be absolutely impossible for one person to do. And that's why you need a machine learning model that will go in and just try an absurd number of combinations and ultimately narrow down the range of possibilities until the model finds something that works quite well. And when I say find something that works quite well, it's found DNA methylation sites that track with age and health and lifestyle or given outcome we care about, that it can combine mathematically into an output that we call epigenetic age. And
I see.
these are usually very simple equations. I mean, the process itself is very laborious for the model to do this, but the equation itself that predicts age is often quite simple. It is a series of methylation values modified by a parameter. And the number of CpG sites you have, determines the number of variables that ultimately get added for the final calculation.
I assume with the millions of variables, the dimensionality is probably a problem, right? That's why you're using lasso to try to penalize a little bit and reduce the dimension somehow.
Yeah. So when you use something like elastic net or lasso, lasso is better for really making the list as small as possible, right? So an elastic net approach is I have all of these variables. I know most of them are not necessary or useful. Please find a good combination that work well. Lasso is more strict than elastic net in that it really tries to use just the smallest number of inputs possible. And that's why lasso models tend to have fewer inputs than elastic net models.
I see. Excellent. So in the end, you end up with this is a kind of a linear model that you are able to present in a way that is interpretable, right?
Yeah, you can interpret it in the sense that the equation is understood. You can share basically the coefficients, the interface values that go into it. You know what the methylation sites are, and you just need to get those values from an array or some other measurement modality to plug in. Now, I would say the field is still growing in terms of making it further comprehensible, right? Of, okay, we now know that you have a delta age, let's say of +5 years or -5 years. A lot of research that's actively going on, and we're part of that research at Tally Health, is to try and understand on a deeper level, okay, why is that happening? And why do we see, what are the methylation sites that are really driving that shift? And is there something about them that's relatable? But this is a really complicated question and a very challenging thing to do. But we are seeing that more people in the field of epigenetics and aging are getting more interested in uncovering these additional levels of detail of why you see a certain shift the way you see.
Perfect. Now that we have these aging clocks, what has been the experience when you apply them to animal models initially? So is there, the accuracy is decent, is something that is applicable, if you will?
Yeah, so we haven't applied them to animal models at Tally because we work exclusively with uh, human methylomic data. But if you look at the broader literature, many clocks have been made for animal models, and several of them I would say have been validated interventionally. So if we look at something like caloric restriction in a typical laboratory setup where the control mice are very sedentary and don't move a lot and gain considerable weight with age, we know that the caloric restricted mice are going to be leaner and healthier and live longer. And we've seen that there are clocks that can capture that difference. There's also been a work done with a frailty based clock that predicts age given frailty index scores that was sensitive to methionine restriction. And there's been other cases as well. And the idea here is that if you can get a clock that's sufficiently responsive to longevity interventions in mice, that you could potentially accelerate longevity studies. Because right now it's very time consuming and expensive to get enough mice for a well designed study and then follow them for years with an intervention. So if we can get to a point where we have confidence in a clock to predict the longevity outcome, then that might be a way to really rapidly assay different interventions in mice.
Mhm. Mhm. And that is very useful. And I assume that that in animals you may have the advantage of a shorter lifespan, and the studies can be completed in a shorter period of time as opposed to humans, where the follow-up is much more cumbersome, right? So that's why these aging clocks are so important. If you are able to apply them to humans, because then you have an instrument to measure the effectiveness of the different maneuvers that you can implement to try to affect the aging process. Am I right?
Yeah, so right now it's basically impossible to do a longevity study in humans without spending an astronomical amount of money and time, right? So let's say we give a new drug to individuals that are 7 years old and we want to track them until end of life. Well, that is just such, you're a medical doctor, you understand how complicated and expensive this would be to do with patients, and it's just not feasible
Hmm.
from the time and cost perspective. So the goal is to find some kind of surrogate markers that can speed this process up. And that's what the field has been pushing towards with developing more powerful aging biomarkers and then comparing different aging biomarkers to understand the limitations of each one and how they can be further improved. But that is a fundamental goal of this whole field, which it's been described as biohorology, the aging clock space, is to really produce biomarkers that can allow us to do a better job learning more about longevity in humans.
Are these clocks incorporating data not related to the epigenome, like lifestyle, nutrition, to try to improve the accuracy of the models?
Yeah, so with the paper we just published in Geroscience, that's a great question you ask. It's called Chic Age. And what we did is we used a very unique approach. We had methylation data for more than 8,000 people, spanning an age range of 18 to 100 years. And we also had digital questionnaire data asking about health and lifestyle. And we wanted to design a next generation epigenetic clock that didn't just capture chronological age, but produced an output that significantly correlated with both health and lifestyle. And we use that by taking a very unique approach involving simulated annealing, which is a probabilistic optimization method. And the result of that is we produced an epigenetic age and found that the delta between that epigenetic age and chronological age significantly associated with BMI, weekly exercise, weekly alcohol intake, smoking status, perceived stress, social satisfaction, the amount of a diet that's plant-based, basically diet quality, sleep quality, and other factors as well. So we were able to produce a novel epigenetic age estimate that is relevant to a holistic suite of factors. And what's cool about that is you don't actually need the survey to produce that, because the model's already learned essentially what methylation sites track with things like exercise or alcohol or smoking. And that's built into the algorithm.
Yeah, that's impressive. And also when I went over that paper, you don't even need blood for that, right? You get the sample from the cheek. That
Yeah,
is
it's
a
it's a
bonus.
buckle. And you know what? It was really fascinating. This was an experiment of, to be candid, can you see these type of associations in a cheek swab? Because previously, these next generation models, many of which are really wonderful, have been built in blood. And blood is a wonderful tissue. It's obviously invasive and not great for people in a home setting, especially if people have a fear of blood or they have blood flow issues. And there's also a certain point where if you're, let's say, 95 and living at home, it might be more challenging for you to do blood collection without help from somebody else. Obviously, it's going to vary in a variety of things. So we really wanted to pursue a cheek swab because it's so easy to do. But no one had done this before. No one had seen if you can see these kind of next generation epigenetic age associations in buccal. So it turns out you can, which is really interesting. And just from a biology of aging perspective, the fact that something like weekly exercise or perceived stress would show up in the DNA methylation markers in your cheek is really fascinating. I think that's really intriguing. And it's not actually obvious that that would be the case.
Yeah.
So
And I think it's a game changer because of all the factors that you have mentioned, and it's awesome. Now, if we move on to the different strategies affecting aging, when you use the aging clocks, so what are the most important lifestyle strategies that have shown increasing that delta between biological age and chronological age?
yeah, it's a wonderful question. If you look at the more recent next generation clocks, the ones that are more relevant to longevity, a lot of what we see is what we'd expect. A lot of people have joked that the best way to age is to do what your grandparents always told you to do, which is to eat your vegetables, to be physically active, to go to sleep, not drink too much, obviously not smoke, etc. But it really is what you would expect. It's these holistic lifestyle factors that are not only really important for our lifespan and health span, but these next generation clocks are picking up on them. So in the paper we just published, we saw that virtually everything we looked at in the survey, every lifestyle factor, was significantly correlated with cheek age. And the ranking we saw was actually surprising. It was not what we expected. And so at the very top, in terms of what was the most significantly associated, this actually was more expected. It's BMI, smoking, and alcohol. That's not especially surprising. But then after that, perceived stress and social satisfaction were actually more significantly associated than exercise and diets, specifically how much of a diet is plant-based, and also sleep quality. So, if you had asked me personally, what do you think is, if I had to rank these, I'd probably put exercise at the top, followed
Yeah.
by diets. But, that's my personal bias and, my own thinking. I also just love exercise. So I want to believe that. And, our clock did find that these are still very important variables, but it was fascinating to see these more psychological variables
Yeah.
like
And the
stress
social
and
interactions, right?
yeah, actually be more significant than exercise and a plant-based diet. And, we need to learn more about why that is. But that is really interesting, especially given the ever-growing body of data really arguing that mental health maybe is not that different from physical health. And the distinction in some ways is fairly arbitrary.
Yeah, I think this is actually golden information because this is something that we need to pay attention to. And you are right. So the lifestyle changes that we recommend and we implement ourselves tend to be focused on nutrition and exercise for the most part, right?
Yeah.
But what you are telling me is that the other dimensions are equally important. And if you want to have a real holistic approach, you have to incorporate them as well, right?
Yeah, and here at Telehealth, we give people action plans. So we're a direct-to-consumer company, and the recommendations we give, we put into 5 buckets, 5 pillars of holistic aging. There's diet, fitness, sleep, mental health, and habits. Habits includes things like alcohol, tobacco, smoking, vaping, things like that. And we intentionally created 5 different pillars because we think they're all really, really important for helping people maximize their longevity. When I say longevity, I mean a combination of lifespan and health span.
And health span.
And if you look, there is really fascinating data suggesting that each of these on their own are really, really important. And if you look at older individuals and you measure their stress and you track them over time, people that feel overwhelmed or that there's a huge amount of stress in their life, they are more likely to die sooner, and they're less likely to make it to a much older age. And this is virtually true of variables in every pillar I mentioned, whether it's physical activity or social satisfaction or something like a Mediterranean diet, which is personally my favorite diet, though I know diets are highly personalized. And so I don't think it's just one thing. I think it really has to be a kind of systems approach where we try and tackle all of these the best we can to maximize our chances of excellent longevity.
Yeah, you're right. And all these factors kind of work upstream, but there are certain interventions that tend to work more downstream, like pharmacological agents.
Yeah.
So, say, obviously, rapamycin has been associated with longevity in animal models, but more important to me is metformin. So, what is your experience when these clocks are applied in the context of using metformin?
Yeah, so this hasn't been thoroughly researched. There has been some data looking at metformin and epigenetic age, and my recollection of the data was that it wasn't significant at the time. But to be fair, it hasn't been really thoroughly studied, and there aren't lots of data sets out there that are compatible with next generation clocks that are just widely available. But metformin is really interesting because it's incredibly effective for patients with diabetes or prediabetes. But there's no doubt that it has this amazing safety record. But we just don't know if it's a good idea for people to take that don't have prediabetes or diabetes. And I know that Dr. Nir Barzilai is still pushing forward the TAME trial, the Targeting Aging with Metformin trial, to really test this, where you take older adults that don't have diabetes or prediabetes, you give them metformin, and then you follow them for years, and you look at outcomes and mortality. So I think that is the kind of test that will really definitively decide, if metformin has a strong role to play for people that have normal HbA1c and fasting blood glucose.
Excellent. Another intervention that people ask a lot about is vitamin D. Understood as a hormone that has pleiotropic effects. It has effects pretty much everywhere, extraskeletal, if you will. So what about vitamin D?
Yeah, I think vitamin D3 is incredibly important. And I'm fortunate that I live in southern Arizona, so I get lots of sunlight, and that's not an issue for me. But when I lived in Minnesota, that was not the case, where it's dark a lot of the time, and you didn't want to go outside for a good chunk of the year. So vitamin D3 has this very interesting history where people were very excited about it for bone health. And then there was a very large clinical trial coming out recently, calling into question its effects on bone health. But then other trials have come out really suggesting a prominent role for vitamin D3 in the immune system. And my read of the data is that vitamin D3 is important for immunity. And I think that it's a very reasonable molecule to try and optimize. Especially if you are living somewhere where there is not a lot of sun.
Excellent, excellent. So, what do you think is coming next in the field of these aging clocks? So, what should be researched better to close the gaps that still exist and that hinder the clinical translation of these technologies?
Yeah, I think what we really need are a few things. We need more validation. So we need to take these clocks and run them in controlled clinical settings to validate them for the clinical community. Because I think that ultimately that's what clinicians want to see, is that a next generation clock is going to provide information that they can't already get. It's going to help them with their decision making or their ability to forecast an outcome for a patient. That will also have the added bonus of teaching us more about what kind of interventions affect these DNA methylation sites and epigenetic aging clocks. And I alluded to this earlier too, but the field is really moving toward trying to understand the inner workings of the clock. Some clocks use thousands and thousands of DNA methylation sites, which is really exciting because then you have more options to work with in terms of capturing different factors. But then there's the added challenge of complexity of how do I understand what it means if these sites change versus these sites. So I would say really trying to offer more clinical validation. And one thing that's great, by the way, is more and more people are sharing data publicly in databases like Gene Expression Omnibus, which really helps facilitate validation for researchers, such as myself and others. But yeah, that alongside with just really decoding what's going on a deeper molecular level.
Yeah, and this is as we discussed at the beginning. This is actually shifting the paradigm and the way we see aging, because it's not only descriptive; it's transitioning to the prescriptive part. And I think the process of aging is something that can be intervened, and having these markers, namely aging clocks, is definitely a very useful tool. And I am sure that the medical community is ready to embrace all these advancements in technology. And I appreciate that you share this information with us. And I want to thank you before we close for your kindness and for sharing your wisdom with us.
Oh, thank you so much. It's been a pleasure, and would be happy to do it again sometime.
Excellent. Thank you so much. For our audience, we are going to put the link to the papers that Dr. Johnson was mentioning and some information about his enterprise and all the nice things they are doing there. Don't forget to leave us the feedback, and see you in the next episode.
Thank you so much for tuning into this episode. If you enjoyed it, please don't forget to give us a thumbs up and share it with your friends and family. Make sure to subscribe to our channel and hit that notification bell so you never miss an episode. If you have any questions or thoughts about today's topic, we'd love to hear from you. Feel free to leave your comments down below. For more information and references related to today's discussion, you can find them in the video description below. We appreciate your support and look forward to having you back for our next episode.